Hai semua. In this particular lesson, we will focus on the proteins, another type of molecules of life that we have to know. Proteins are organic molecules, meaning that they consist of carbon, hydrogen, and also oxygen. And another type of atom that is also included in the proteins will be the nitrogen. Proteins are also called polypeptides, in which this molecule consists of many monomers which are known as the amino acids. We will now focus on the monomer of the proteins, and this particular diagram shows the basic structure of the amino acid. Normally, there will be a carbon atom in the middle of the molecule, and at one particular end, there will be a special type of functional group known as the carboxyl group, the COOH. On the opposite end of this particular molecule will be another functional group known as the amino group, which is the NH2. This amino group is having a basic property, while the carboxyl group will be having an acidic property. And that is why we call this particular molecule as the amino acid. Apart from the carboxyl group and the amino group, there will be a particular hydrogen atom, and on the opposite side there will be an R group, which is actually referring to a side chain of the amino acid. This particular side chain can be anything. It can be a simple hydrogen atom or something that is more complex. There are actually 20 common amino acids that have been identified by the scientists. All the amino acids have the same basic structure. However, they can be differentiated based on the different type of side chains. And so we can actually classify the amino acids into 4 major types. The first type will be the non-polar amino acid, in which this particular type of amino acid will be having a hydrophobic or non-polar side chain. For example, will be the glycine, which is having an H atom as the side chain. The second type will be the polar amino acid. For example, the serine. For this particular type of molecule, the side chain is hydrophilic and has a polar characteristic. Type number 3 will be the acidic amino acid, in which the side chain will be negative charge, or we can say that the side chain will be having the carboxyl group of COOH. Example will be the aspartic acid. And the fourth type will be the basic amino acid, in which the side chain will be positive charge, or we can say that the side chain will be having the amino group. One example will be the lysine. So we should understand that proteins or the polypeptide consists of many monomers which are the amino acids, and maybe we should start with how 2 amino acids can be joined together. In this particular diagram, we can see 2 amino acids joined together due to the process of condensation, in which water molecule will be removed. As a result, a structure known as a dipeptide will be produced. Due to the removal of water molecule, a peptide bond will be joining the 2 amino acids together. And if we want to break down this particular dipeptide, the process of hydrolysis need to occur, in which water molecule need to be added to this particular structure, and the water molecule will break down the peptide bond, forming the two amino acids again. A protein has a specific three-dimensional conformation with certain levels of protein structure. It might be the primary structure, the secondary structure, the tertiary structure, or the quaternary structure. So what is actually the primary structure of a protein? The primary structure shows a unique sequence of amino acids that are joined together by peptide bonds. For example, the primary structure of the glucagon, which is a protein type of hormone, shows a sequence of 29 amino acids joined together by the peptide bonds. So there will be no other extra bonds apart from the peptide bond. Meanwhile, the secondary structure will be shown when the polypeptide coils or folds. This is due to the hydrogen bonds that we can see connecting the monomers together over here and over here, or somewhere over here. Result, there will be two forms of secondary structure that can be observed. The first form will be known as the alpha helix structure, which is showing a coil structure like this. And the second type will be the beta pleated sheet, which is showing a folded type of structure like this. Example, the keratin. Keratin is a type of protein that shows the alpha helix structure. And sometimes the polypeptide chain may be coiled even further to form a globular shape. And this is called the tertiary structure. The tertiary structure is maintained due to certain bonds or interactions between the side chains, such as the disulfide bonds, the ionic bonds, the hydrogen bonds, or the hydrophobic interaction together with the van der Waals interactions. Example, the myoglobin is a type of globular protein that will be showing the tertiary structural level. In this particular diagram, we can see how certain bonds can maintain the tertiary structure of the protein. Over here, we can see the disulfide bond, which is created between the two sulfhydryl group. And over here, we can see the ionic bond, which are strong bonds created between the positive and the negative charge side chains. Over here, we can see the hydrogen bonds, which are actually weak bonds between the polar side chains. And over here, we can see the hydrophobic interaction, which is actually a weak type of interaction between the non-polar side chains. Sometimes, a protein can show the quaternary structure. In this particular case, one particular protein molecule might be having two or more polypeptide chains. All the chains might be joined together by certain bonds such as disulfide bond, ionic bonds, hydrogen bonds, or even the hydrophobic or van der Waals interactions. For example, the hemoglobin, which is a very important type of protein which can be found in our red blood cells. The hemoglobin is actually a type of protein that is showing a quaternary structure because it consists of 4 polypeptide chains. If you look at this particular diagram, we can see that one hemoglobin molecule consists of 2 alpha chains and another 2 beta chains. The structure of the protein can actually be affected by pH and also temperature, in which extreme pH and extreme high temperature can cause denaturation to occur on the protein, in which the protein will be changed due to the breakdown of certain bonds such as the hydrogen bonds, the ionic bonds, the disulfide bonds, and also the hydrophobic interactions within the original conformation. As a result, the protein loses its original conformation, and finally, the protein will lose its ability to function. In this particular diagram, we can see the original protein change its conformation due to the process of denaturation, and the denatured protein will not be able to function as it should be. Finally, we need to know that proteins can be classified into three types: the fibrous proteins, the globular proteins, and also the conjugated proteins. Hopefully, you can refer to your notes and try to find differences between these three classes of proteins. And with that, I end my lesson for today. See you guys later. Bye-bye.